Hey guys, it's Miss Lebo. Today's lesson is going to be about Gregor Mendel. We're going to find out who he was, why he was important, and then how he overall influenced the topic of biology. Of course, we're going to start with your objectives for the lesson, so I would pause the video, take a look at your two objectives. The first one has a lot to do with different terminology for this lesson, and so I would jot those down maybe in the sidebar of your notes just to make sure that you are able to hit each one of those different definitions. And then the second objective is going to be addressed later on within the lecture. These are important. These are directly connected to your unit overall exam, and so if you're able to correctly define these and talk about these, then you should be in good shape. Okay, so let's get into it. First off, we're going to talk about Gregor Mendel, and he was an Australian monk, and he worked in the gardens of the place where he lived, and so he is known to us as the father of genetics, and this is because while he was working in his garden, he was looking at pea plants, and he got really interested in how these pea plants changed from generation to generation. When we talk about the father of genetics, we have to define what genetics is. And so genetics is the scientific study of heredity. Mendel was able to study these pea plants by looking at their different traits, and he performed experiments on these pea plants. They were a really good model for him because their traits were going to be differentiated in a very simple manner that he was able to tell the difference between the different seed colors, the different seed coat colors, how the coat was shaped, and things like that. Before we go into talking about his experiments, we need to know a little bit about how plants are going to reproduce. And so the peas that he was using, they were normally going to reproduce asexually. And so what that means is that these plants were normally going to self-pollinate, and then that's going to produce offspring that are identical to the parents. And so there was no genetic diversity between the parents and the offspring, and this is what we call true breeding. That's a definition that you need to know, so I'd mark it down and make sure that you're able to explain it. So what Mendel did within his experiments was that he was going to take the pollen from one pea plant and he implanted it into another pea plant. So that means that these plants were reproducing sexually, and then this is what we called crossbreeding, because we were crossing and implanting from one plant to another pea plant. So why is crossbreeding important? I touched on this a little bit in the previous slide. We are going to need to crossbreed our plants so that we're able to look at the genetic diversity. If we were just using the true breeding method, again, that is going to be identical offspring to the parents. And so crossbreeding is going to make sure that there is a variety of characteristics from one individual to another. And this is what we now know are known as traits. And so the different traits that Mendel looked at within the peas were seed color, seed coat, seed shape, plant height, and many more that we'll show you later on in the presentation. So these traits were really good examples within the pea plants because you either had one seed color which could be green or another seed color which could be yellow, and there was no other option. So because it was a 50-50 chance, his experiments were going to be very easily manipulated and his results were going to be very easy to see. Also, while he was doing his experiments, he needed to be able to name the different generation of plants. And so this picture on the side is going to show you how we define the generations. So we start off with your parent generation, the parental generation. We also will label that with a P. This is going to be your first generation of plants. This was the initial crossbreeding that he did, so when he was taking the pollen of one plant and implanting it into another pea plant. So this was the very start of his experiments. Those parental generations gave rise to the first filial generation, or we call that F1. And these are going to be the offspring of the parental generation. After that, you can continue your experiments and you can use the F1 generation to give rise to a second filial generation, and we denote that as F2. So that means that overall we start with the P for parental, you go to the F1 generation, first filial, and then below that you could go to the F2 generation, which means second filial. All right. So he was doing his crossbreeding, he was working on these different experiments, and you can see along the side of the chart, you have a trait that he was going to be experimenting with. And so the first one, we're talking about plant height. So what Mendel did was he took parent A, which their trait was going to be a tall plant, and crossed it with plant B, which is going to be a short plant. Those parentals are labeled as the P generation, and then in the very last column, you see what their offspring were showing for the trait. And so that means that when he crossed the tall plant with a short plant, the offspring were going to be showing the tall trait. This was true for all of the experiments that he performed with the tall plant and the short plant. And so then this is going to lead into what we know as dominant and recessive genes. Again, the same thing was repeated for the seed color, which is going to be the next row down. And so he took parent A, which was green, crossed it with a parent B, which was yellow, and that gave rise to offspring which were green. Another example was that he looked at seed shape. He crossed a round with a parent who was wrinkled, and then that gave rise to offspring which were round. These offspring were a result of crossbreeding, which we know is going to also be called sexual reproduction, and so then these are going to be called hybrid plants. So overall, this chart off to the side is going to show you the different traits that Mendel looked at, and you can take a look to see after the end of his experiments which traits he labeled as dominant and which ones were recessive. It's important to note that he took a very long time to make sure that all of these experiments had multiple trials, and overall he found that the dominant trait for each one of these different categories was always going to show up again and again and again. So for example, the first trait that we have is the shape of seeds, and so Mendel found that the dominant trait was always going to be round instead of wrinkled. So now we can get into some conclusions. The first conclusion is that biological inheritance is passed down through generations by what we were calling at the time factors. There are air quotes around the term factors because this took place a long time ago, and Mendel's experiments came in history before the uncovering of DNA, and so we weren't able to quite tell that these factors were within DNA, but we did know that there had to be some type of deal with inheritance. So Mendel is known as the father of genetics because he was the first one to talk about any type of inheritance, dominant, recessive genes, traits, anything like that. All of Mendel's work led up to what we now know about DNA and genes and alleles. So what we are now able to tell is that we have genes and alleles, and these are going to work together to give us our different traits. So the gene is an inheritable factor that determines traits. But an allele is a different form of a gene that can provide different expressions of a trait. That's a lot of words, so we can break it down into an example. A very simple example on our human bodies would be earlobe shape. So everyone has a gene for an earlobe shape. So that means that every person is going to have a shape for their earlobe, but the allele is going to be whether your earlobe is attached to the side of your face or if it's unattached. And so the allele is able to be different between people, but everyone needs to have a gene trait for your earlobe shape. An example for the pea plants was that the gene would be seed color, and then between different plants, the allele would be that you could have a green seed or you could have a yellow seed. Two more conclusions that are important to our overall concept within biology is that we have a principle of dominance. And so most of us are very familiar with this. This is whether an allele with a trait can be dominant or recessive. Dominant traits are always going to be expressed within an individual. Recessive traits will be expressed only if a dominant allele is not present. Underneath the text, it says that dominant traits are written with capital letters and that recessive traits are written with lowercase letters. This notation is going to be important when we move on to working with Punnett squares in our next video. And then the third conclusion is that we're able to describe gene pairs. So we can call gene pairs either homozygous or heterozygous. So these prefixes are coming up again. So remember that homo means same and hetero means different. So for the homozygous gene pairs, this is having two of the same allele for a trait. So you could have a dominant trait for, we're using the letter T for tall, a tall plant. You could have a dominant trait from your mom, a dominant trait from your dad, or to be homozygous, you could have a recessive trait for the tallness, which would be a short, from your dad, and a short trait from your mom. On the opposite end of that, heterozygous would mean having two different alleles for a trait, so you could have a tall trait from your mom and a short trait from your dad. And then one more conclusion. All of this together, we're going to be talking about genotype versus phenotype. So the way that I remember this is that your genotype is going to be the genetic makeup of a trait. So the genetic makeup is going to be whether you received a dominant trait from your mother or if you received a recessive trait from your mother, and the same thing goes along for the father's lineage as well. So that's the genetic makeup that's inside of your body. And the phenotype is going to be the physical expression of that trait. So that's going to be what you show on the outside. You can see the table at the bottom, and that's going to show you genotype, which are the letters, the phenotype, which is what you show on the outside, and then you can practice your gene pair notation to tell whether it was homozygous or heterozygous. All right, so our last question. Mendel noticed that the recessive traits would reappear in the F2 generation. So if you look at the picture on the side, we're starting with the F1 generation, which is very important. We are not starting with the parental generation right now. So you have the F1s, and you're going to cross them together. Their chromosomes are going to separate during gamete formation. And remember, we are using gamete formation because this is meiosis. So these gametes are going to have one allele for a trait within each of their cells. And you can see that in the middle row of cells within the picture. And then at the bottom, they're going to come back together during fertilization. And then if you received a short trait, recessive trait from your mother and a recessive trait from your father, then that overall recessive trait is going to show up in the F2 generation. Okay, so picking up where we left off, we were talking about how the recessive trait is going to reappear in the F2 generation. And so this starts in our F1 generation. So remember, we're not starting with the P. So we have our F1 generation. They are going to split through segregation. The chromosomes are going to separate, and then those gametes are going to form. Once those gametes are formed, they are going to have to come together, one from the mother, one from the father, through the process of fertilization. And when that happens, there's a possibility that you would receive a recessive trait from your father and a recessive trait from your mother. If that happens, both of those recessive traits come together within the F2 generation, but then and only then the recessive trait is going to be shown within that individual. So this is how we see the recessive trait reappearing within the F2 generation. So now we're at the end. This slide is included just for your benefit. This is a slide where you can practice summarizing some of the main topics within this lecture. There are 3 examples across the top: dominance, gene pairing, and segregation. Again, these are just examples. If you're able to write a quick 1 to 2 sentence summary about these without looking at your notes, then you are going to have them down pat and you're going to be ready to talk about them in class. If you're struggling a little bit, then I would maybe go through, look at this note section, maybe re-look at the video a little bit. And of course, you can add on to this. So if you want to practice summarizing any other topic within this video, you can do so on this slide. And of course, if you have any questions going through the lecture, or if you want to review anything in class, jot down those questions on this slide too, and then we can come back to them in class, and you can turn to your classmates, you can turn to a teacher, and you can ask those questions and remember the questions that you had while going through this lecture. Of course, we're going to end by saying that you should review the objectives, which were at the very beginning of this video. If you're able to go through the objectives and check them off, then you have done your job for this video, for this topic, and you're ready to go in class. So thanks for watching. Ask any questions in class, and we'll see you next time.